there is a problem. And I mean an excused absence type of problem. You're in the hospital, your car breaks down, something. Okay? The only other options you have, and you have to talk to me before you do this, I do teach the general biology course. There's a 7 a.m. lab. Yeah. So, that's your only other option on here. So, try to be here on time. Alright. Let's see. Okay, next portion we're going to get into the microscopes, and that's pretty much the last couple of labs. There is... Oh, crap, they didn't put out our cell models. Okay, if y'all need to see the cell models again, they're up in the thing. We'll get them out here in just a minute and put them up out front. Okay. For the microscope here. Do I really need to say anything about it? We took that one day and yeah.
Do you know
The parts and the functions of the parts. So what they usually do is there's like a little tape arrow somewhere on this microscope, and they'll go, what is that part? And then the next question almost automatically is, what is the function of that part? Try to keep the function down to like just a couple words, like 2 or 3 words, and you'll be good. One of the ones that tends to end up on here a lot, you'll remember the diaphragm down here? Yeah. That one tends to be on there a lot. If you need to answer that one, go through and just say, it adjusts the amount of light. Okay. Not going to go over that too terribly. Most of the other slides that they have, if you've got your review with you, they go through a listing of all the parts and pieces that you need to know of it. If you need to, everything's set up. All the wet mount slides are set up. Let's see. Remember, the only prokaryote that we looked at this semester was the Anabaena. That was that blue-green bacteria. And that's that one that looks like the string of pearls. That's the only prokaryote we looked at this semester. I just repeated myself twice, so that's probably going to be a question on the test.
The
Anabaena. All the rest of the cells that we looked at, they're all eukaryotes. No, only prokaryote was Anabaena. Alright, the rest of these, I'm going to let y'all just go through them. If you need to, take pictures, whatever. Now, here's one of the things that I usually tell the students. If you are in here in the lab practical, okay, and you've got the microscope and the slide sitting in front of you, have I said anything that you cannot look at the slide physically? No. Remember that a couple of the wet mount slides that we did, like the onion, the potato, the carrot, even the flower petal, have very distinctive slides to them. You can look at the slide and tell it's a flower petal, correct? Yeah. Okay. If you need to do that to help jog your memory of what's in it, do it. Okay? Yes?
Are they all going to be wet mount slides, or are there some of them?
Some of them are wet mount, some of them are prepared slides. What I will tell you, the general rule is if it's anything like the little unicellular organisms we looked at that were flitting around the slide and it was hard to catch up with them, most likely those wet mounts won't be out here. It will most likely be a prepared slide. But for the plants, those are very easy to make wet mount slides. Those usually end up being out.
And if we have trouble finding it, we can call you over.
Yes. Do not try to adjust anything in here on the test. If you think anything is out of focus, out of whack, the pointer's been moved, it's been bumped, don't even try to do it yourself. Just go, hey professor, can you come adjust? And I will do this for every one of them. Now, the caveat on that is, if you don't ask for help, and you don't ask for it to be readjusted, and then you get back your test and you missed a question because the pointer has been moved to another structure, don't come crying to me. Okay? It's up to y'all to ask me at the time of the test to adjust these things. Yeah? Default, if it looks like it's been bumped or moved, or it's not clear that the pointer is directly on a structure, ask.
Well, what if it went from the
doesn't matter. You can ask me to readjust every microscope slide as we're going through. I don't mind, I don't care. I will continue to do that for everybody. The only thing I would say is if you see me adjust this one and you're at that one and move over, odds are it's probably still where it needs to be. If we can just get y'all to, whenever you're using these microscopes during the test, don't even touch anything. Just look in the oculars. Don't even try bumping the table. Just lean over, look in. Most of them won't be out of adjustment. Okay? Also sat down last week with the lab supervisor, and a lot of these microscopes, y'all know how we were having a problem last week if some of them had the stage dropping, others of them had the 40X where you couldn't see through them, that kind of thing. Every one of these still in here have been broken apart, re-put back together with new parts. So as far as I know, everything's still working in here. If you find something today, let me know. We will tear it apart again. All right.
So these are set up just like they will be on test day.
Pretty much. The only thing that will be added here is a card right next to them asking you the 2 questions for that station. Matter of fact, there will be, like the station on the measurement up there, where you use the smaller ruler, that's set up exactly like the station would look. Let's see. That should take care of 5, 6. Now we just need to get to 7, which is the pH lab. Now, if y'all remember last time, or Monday, remember I told y'all these would be the only ones that you actually have to do something on? Well, this here is set up like the station would be on the test. Okay? If you would. Now, if you don't know what's in the test tube, should you be messing with it too much? No. Because there's a possibility it could be a heavy acid or a heavy base. So, if you would, make sure when you're testing these things, take your litmus paper, set them on the paper towel. You only need a small drop, and then put it back. Small drop, put it back. Alright? Once you've done your readings, please pick up your litmus papers. There will be a small beaker that says trash next to it. Throw it away. Don't trust anything that's left on here, because some people tend to, they'll put the litmus paper and then test cross, so you don't know which test tube it goes through. Do your own test. Alright? Like I said, this little card here is probably going to be this card sitting out for you. You don't have to memorize the colors, just know how to read from it. Now, here's the weird thing. Okay, this one says here, for this buffer station, which one, test tube A or B, has water, acid, and a buffer? Kind of like we did for the lecture exam last time. What's the most important things you're looking for on that question there?
Water.
Water? Not so much. What's the other 2? Acid and buffer. What do buffers do?
Stabilize the pH.
Stabilize the pH. So when you test these, are you going to expect an acidic pH or a neutral pH with the buffer?
Neutral.
Neutral. Okay, same thing on the next one, you're only looking at acid. They kind of just put the water on there to be complete on what's in the test tube, but the water itself is not going to affect your test. So disregard water or anything like that. Look for the things that we were testing for. Okay. Let's see, what else? The other thing that you can do is, like on this one, I think there's a, if I remember correctly, they put an acid in A and a base in B. And then they just go through and it's like, what is the pH of each test tube? When you're looking at this, oh my God, your professor has butterfingers today. Okay, when you look on here, which one is this going to be? Closer to 11 or 12? 12. Now, understand, because I know that this is a judgment call on coloration, you know, if you say 11 or 12, I'm probably going to take it. But if you say like 9, and it's a difference between a green and a blue, that's probably wrong. Okay? So get it as close as you can. Same thing with the acid. Like here, it could be probably 1 or 2. So I'd probably take either or, but if you said it was like neutral, yeah, that's not gonna work. But again, after you do your testing, just throw it away. Let's see, antacids. What was the pH indicator I had y'all repeat several times?
Something purple.
Bromocresol purple?
There you go. Do y'all need to say it like 50 times with me? Bromocresol purple, bromocresol purple. Come on, get it into your head. Bromocresol purple. I can almost guarantee that's going to be a question. Now, on the antacid here, which of these two, test tube A or B, would be the better antacid?
A.
Why is that?
It's a purple, so
Yeah.
it's D.
Yeah, so if you remember what we were doing, we were adding acid until it overcomes the buffer, and that's whenever it turns yellow. Okay. Yellow substance. So which one, A or B? A was the purple one, B was the yellow one. Which one had the lower pH?
A.
I heard
B.
confusing, I heard both A and B.
B.
Which one, A or B?
B.
B. And you can tell that because it turned yellow. Yellow indicates a lower pH, or a pH of, usually it's 4 or below. Anyway, honestly, that's pretty much the test. So, my suggestion, and I'm going to tell you all this honestly, okay? Those people that usually make the better grades on the lab practicals are the ones that stay today until the end of time, making sure they know each and everything. Usually the people that leave the soonest are the ones that make the lowest grades. All right? So spend your time wisely. Yeah?
Is open lab set up like this too?
Yes, Open Lab is going to be set up exactly like this. It's on both Friday and Saturday mornings. On Friday mornings, it's from 8 to noon over in the this lab right next to us. And on Saturday, it's from 8 to 11:30, and it's over in the other hallway in lab room number 2117.
And there's people you can,
It is already posted. It's one of your announcements.
and
Yes.
there's people that can help you, right?
They have lab techs there. On Friday, there will probably be at least a couple of us full-time professors there, but on Saturday, I can't guarantee that. So Saturday will be mostly just open for y'all to study. So anyway, go through, take pictures, make drawings, do whatever you need to learn the information.
So for